All right, so our learning unit on enzymatic catalysis. So thinking about enzymes catalyzing reactions. So we begin this with kind of a review of what it means to think about reaction coordinates. So again, that was sort of a skill we had in our fundamentals unit, but that was really important to prime us to think about how enzymes are going to be able to catalyze reactions. We kind of have a little bit of an explanation of the different ways that enzymes can kind of catalyze reactions. And then we're going to actually learn about 4 different enzymes. So 3 are shown here, kind of hydrolyzing our biopolymer linkages, if you will. So phosphodiester bond cleavage by RNase A, glycosidic bond cleavage by lysozyme, and then serine protease cleavage of peptide or amide bonds. One more enzyme that we are going to study is carbonic anhydrase, just because we've spent so much time sort of thinking about that as a model enzyme. So reaction coordinates, we're actually going to see this reaction coordinate one more time in our kinetics chapter, but make sure that you understand sort of what's going on here. What is delta G? What is delta G double dagger? So I'm going to walk that through you again for you here. So again, this just shows simply we've got reactants being converted into products. The difference in energy levels between reactants and products is delta G. So again, my line isn't exactly perfectly straight here, but the difference in energy between reactants and products is delta G. Delta G we think about as how much and whether. The how much comes from the magnitude, the actual numerical value of delta G, because we can use that to relate to an equilibrium constant, and equilibrium constants are concentrations of products over reactants. So at the end of the day, the magnitude, the value, the size of delta G is directly proportional to how much reactant and product we have. The whether part has to do with the sign. So whether a reaction occurs, whether it's spontaneous or not, has to do with the sign of delta G and whether it's positive or negative. So again, a negative delta G indicates a spontaneous reaction going downhill. If we had the products higher in energy, that would be a positive reaction, not spontaneous. We're going to see lots of examples of, when we study metabolism, of coupled reactions where one reaction might have a positive delta G, but it's pulled forward by a subsequent reaction that has a negative delta G. So again, delta G is whether and how much. Delta G double dagger is how fast. So again, this really only has a magnitude portion to it, and the magnitude really has to do with how fast it is. So a large delta G double dagger is a slow reaction because that represents a large hill that has to be gone up and over. You determine delta G double daggers by the energy difference between an isolatable species that lives in a valley, like reactants here or even products, and a transition state energy. So it's always going to be an uphill arrow. And enzymes make reactions faster by lowering the transition state energy. So when you make delta G double dagger smaller, you make a reaction faster. You have a smaller hill to get up and over. So how this happens is enzymes work often by finding some way to stabilize the transition state. So we talked about this in class. One of your homework problems actually has you write 3 times the fact that when you make intermolecular forces or bonds, right? We make energy or release energy, but when you're going to break intermolecular bonds, that's when we take energy. That's when we go uphill. So if we are making new intermolecular forces, that's always going to be stabilizing. That's always going to be lowering in energy. And because we're lowering something in energy, first law of thermodynamics says we cannot create or destroy energy. We're just going to convert it into other forms. That energy has to go someplace, so that energy is released. So anytime we make intermolecular forces, we are releasing energy. So a big thing to remember here is that enzymes do not affect delta G. We don't take a non-spontaneous reaction and make it spontaneous, and we do not change the amount of product. So we don't change the whether, and we don't change the how much. We just make it quicker to get to that same distribution of product. So a few slides here that were rather busy as we started out our chapter, just kind of giving us the bird's eye view of what's gonna happen here. So these are some of the questions that you'll see on your exam, sort of like the nuts and bolts, making sure that you understand that lysozyme is an enzyme that's going to break glycosidic bonds. So some of your homework problems really sort of reinforce that, thinking about what the polymer substrate is, what the linkage is, what the enzyme is, and sort of a little bit of detail about where that enzyme is located in terms of what it does. So all of the reactions that we're going to see, even the carbonic anhydrase one, involves water. So the polymer hydrolysis reactions really are using water to break apart a linkage. So again, the linkage in our carbohydrate polymers is glycosidic bonds. So we're going to have lysozyme that does that. DNA and RNA having phosphodiester bonds are going to be using RNase A. And I should say RNase A is really just for RNA here. Protein or amide bonds are going to be cleaved by serine proteases. And big picture, why is something like this important? A lot of these enzymes that we're going to talk about are ones that live in your gastrointestinal tract. Remembering that only monomers can be taken up from your GI tract and delivered into your bloodstream. If we have consumed fuels that are polymers, we need to have ways of breaking them down into their monomeric form to allow them to exit the GI tract. So amino acids are going to be able to help in the active site of these enzymes do some chemistry. So all of the times when we did our reaction mechanisms and I told you, no, we can't just show PT for proton transfer. I want you to show me the addition or the removal of protons. That's because we're going to map now on enzyme active sites where there can be amino acids that can help. So again, to add or remove protons, we need to have residues that have reasonable pKa values. So again, you need to be able to know your single letter codes. So we've got our acidic residues, our glutamate and our aspartic acid. We've got histidine, we've got lysine, and we've got arginine. So we've got our 5 kind of charged, either basic or acidic amino acids. Note here that these are going to be strange letters. That's why they're hard to remember. But remember, the prevalence of an amino acid dictates whether it gets sort of the starting letter of its name. So the fact that these are the strange letters reminds us that these are not found very often, and that's significant, right? Because nature is just not going to throw these really important amino acids that can participate in chemistry just all over the place. So when we think about stabilizing by providing a charge, it's going to be these same 5 residues. The acidic ones, which are going to be negatively charged, so they can stabilize something positively charged. The positively charged basic residues are going to be able to stabilize something that's negatively charged. So again, in this case, if we're talking about stabilizing something by providing an opposite charge, that is a way that we can decrease the transition state energy, stabilizing that. Another way that amino acids can help is maybe they can act in a nucleophilic capacity. Maybe they could act, if we're doing a lot of hydrolysis chemistry, maybe they could act like they were water, like a hydroxide group. And serine and threonine are going to be what are capable of doing that, right? Serine and threonine having hydroxyl groups. So just highlighting here, serine proteases are called serine proteases because we have a catalytically active serine in the enzyme active site that acts as a surrogate to water and actually does the initial chemistry on our substrate. So a lot of enzymes that do chemistry, and if we think about just our basic fuel metabolism equation, right, we're going to be oxidizing fuels. So clearly we need to be thinking about doing redox chemistry. When we think about doing redox chemistry, the amino acid side chains are not really equipped to do that. They don't have the functionality that's going to allow for that. That's where we see coenzymes come in. Coenzymes really can be thought about as the teeth for enzymes, right? So coenzymes are things like NADH and FADH2 when we think particularly about the redox chemistry that happens with this fuel equation. But again, these guys really are important because they're some of our B vitamins. So one of the things that we introduce in this chapter, and that's going to become important for your last 2 fundamental tests that we'll have after this next exam, is going to be on vitamins and then hormones. So important things. Again, your fundamental tests were on functional groups, we're on amino acids, we're on vitamins, and then are going to be on hormones. And so those are kind of some big picture things that we want to make sure that we are well equipped to sort of think about and talk about. What we're going to do now is we've got 3 slides here that are going to highlight how enzymes work by looking at the uncatalyzed reaction and kind of mapping things on. So I know these slides were really busy and we went through them fairly slowly in class. I'm going to try to go a little bit more quickly since this is just a review. But what we have shown in black in all of these slides is the substrates that we're going to be doing chemistry on. Sometimes we're actually going to be drawing out the actual substrate like we have here with a little dye dinucleotide of RNA. Sometimes we might be showing what we kind of showed on our reaction mechanisms, so very generic using R's. But at the end of the day, what we're going to see here is some important things that I want to highlight. In blue, we're going to kind of show here that anytime we had protonation or deprotonation events happen, those represent positions that maybe we could have an amino acid that's going to serve in one of these addition or removal of proton capacities. So that's why it was important to think about drawing those bases or drawing those acids out, because anytime we show one of those, that represents, in this case, a place where a general acid could help. In this case, where a general base could help. And the reason we use general is we're just saying that we're not identifying who it is at this point. We're saying that that could be sort of a general base that's helping here. Once we get into the enzyme active sites, we'll actually see that there are specific amino acids that kind of operate in this function. So the other thing that we have here shown in purple is 3 steps that are really important that we need to think about with every enzyme active site that we look at. The first thing that we need to think about is that an enzyme active site must perform chemistry on the substrate. So for all of these hydrolysis reactions that we're going to be looking at, that's basically cleaving whatever the linkage was. So in this case, cleaving the phosphodiester bond. The second thing that we need to make sure that we do is use all of our reagents. And generally in this case, that means we're doing hydrolysis reactions, so we need to make sure that we see and use water in our enzyme active site. And then importantly, we need to make sure that the enzyme is returned to its active state. It doesn't really make any sense to have an enzyme just do chemistry once and then not be able to do it again. That's really inefficient. So this is also what we refer to as kind of turnover when we talk about removing products from the enzyme active site so it can do its chemistry again. But beyond just making the seat available for a new substrate to bind, we need to make sure that all of the amino acids are returned to their proper active state. So looking at what we have here again with RNA hydrolysis happens because we have this built-in internal nucleophile that can nucleophilically attack the electrophilic phosphorus of our phosphodiester bond. Again, we're not drawing out full mechanisms here, but we'd form this pentacoordinate intermediate, which would decompose, and then we'd have departure of this 3' portion of our nucleic acid, and we would generate this cyclic phosphate intermediate, 2', 3' cyclic phosphate intermediate. At this point, we'd have our downstream RNA, our 3' RNA, would diffuse out of the enzyme active site, and water would be able to diffuse in. But even just after this first step, we've really only done chemistry on the substrate. We haven't yet used water. And if we thought about the fact that an amino acid might be helping here, and in this case acting as an acid to protonate our leaving group here, or acting as a base to deprotonate this 2' hydroxyl, they're not going to be in the right form. This guy's going to be protonated when he needs to be deprotonated, and similarly this acid down here is going to be in a deprotonated form. But what we can see is a common theme that we see here with our enzyme active sites. We sort of see 2 acts to our play. In the first act, we're going to see chemistry on the substrate, and oftentimes it's some type of an internal nucleophile that sort of helps with that chemistry. Water's going to come in the second act, and water's going to do 2 things. Not only is it going to help regenerate the enzyme active site, but it's going to finally kind of satisfy the fact that we're using all of these reagents. So water kind of comes in the second act of the play here. Usually it will, by its nature, regenerate the enzyme active site, but it'll in this case regenerate or generate our final product, which is our upstream nucleotide substrate, which can also then diffuse out of the enzyme active site. So I know that was a lot to kind of highlight there. Hopefully it made sense that this is a little bit of a review. But again, we're going to be seeing a lot of these hydrolysis reactions, so we're going to need to see water as a reagent. We need to make sure we use that as a reagent. Most of these are going to show chemistry on the substrate happening in the first act before we've even used water. But then water is going to come in to be not only utilized because we need to use it, but it's going to be responsible for regenerating the enzyme into its active state. So another one of our linkages that we need to think about kind of hydrolyzing is a glycosidic bond that we'd have in carbohydrates. So in this case, we're not actually drawing out carbohydrates, we're actually drawing the acetal or ketal. And again, an acetal or ketal is going to be determined by whether this is a hydrogen or another R group that's there. So again, when we have our acetal here, remember this represents our cyclized sugar. So this is the anomeric bond. This is how we've cyclized our sugars. How we polymerize our sugars together is through this glycosidic bond. So the glycosidic bond is responsible for polymerization. The anomeric bond is responsible for cyclization. So again, some of these things are going to be those small details that you'll see on your in-class exam, kind of thinking about the bonds that are responsible for either polymerization or cyclization. The scissile bond, this might be the first time that we've said this term here, the scissile bond refers to the bond to be cleaved. So if we think about cleaving this glycosidic bond, how we did this in our reaction mechanisms is we protonated this group to make it a good leaving group. We had an internal nucleophile that pushed it out, and then we had water come in, react with this very electrophilic intermediate here to generate our final product. Glycosidic bond has been cleaved. We now just have the hemiacetal or the hemiketal, again likened to the cyclized sugar. Same thing that we see here. These three steps need to occur. In the sort of the first half of this play, we saw chemistry on the substrate. Again, by protonating this guy to make him a good leaving group, we could have an internal nucleophile push it out, and we've generated our cleaved glycosidic bond before we've even involved water. Water again is going to come in. It's going to help regenerate the enzyme active site, because if this residue acted as an acid in the first step to protonate this guy to make him a good leaving group, he can be regenerated in his active form by serving as a base in the second step, and that also helps increase the nucleophilicity of water to attack this electrophilic intermediate here, and again generate our final product. So we're going to see some chemistry here for lysozyme that allows us to understand how this chemistry works. Last sort of hydrolysis reaction is peptide hydrolysis. So again, in this case, we did actually draw out a little peptide bond here. So again, having this peptide bond here, we're going to see in our uncatalyzed reaction, what we really showed here was maybe deprotonation of our water to make it a better nucleophile. Nucleophilic attack on this carbonyl carbon generates a tetrahedral intermediate. Tetrahedral intermediate can decompose, protonate our leaving group, and we're going to generate our cleaved product here. So one of the things that we want to think about is, and again, that's just looking at the black and the red that we have there. What would happen if we didn't see water early on here? What if we saw a water surrogate? What if we saw serine? So let's map through here. What if this was not an H2O residue, but if it was a serine that has a hydroxyl group? Nucleophilic attack and generation of a tetrahedral intermediate would now have a serine that's covalently attached to our substrate here in this tetrahedral intermediate. We'd still need to see protonation of this leaving group here, but decomposition of the tetrahedral intermediate will now have not a hydroxyl group here, but serine in our enzyme that's covalently attached to the N-terminal portion of our peptide. C-terminal portion could then depart, and at this point we're left with an upstream N-terminal peptide that's still covalently attached to our enzyme. The C-terminal peptide can depart. Now at this part, we don't really have it shown here on this uncatalyzed reaction, but we need to actually repeat this sequence of events, now having water come in to act as a nucleophile, attack this, generate a tetrahedral intermediate, decompose that, and now allow serine to leave. That's what we need to do to not only involve water, but also regenerate the enzyme active site. So serine proteases are going to be one of the more involved sort of enzyme active sites that we're going to see, because we actually have to do this process twice. We've spent a little bit of time sort of talking about some general aspects of how enzymes and substrates interact, and this is going to lead into kind of our understanding of kinetics. So again, enzyme plus substrate are going to interact to make an enzyme-substrate complex. That enzyme-substrate complex does chemistry, where we're going to generate an enzyme product complex, and then we have to release product here. So substrate binding sort of occurs in this first domain here, and then product release occurs here. So the lock and key model is a very idealistic way to sort of think about the fact that enzymes are very specific for certain substrates. They don't just kind of randomly go around and find anything and do their chemistry on it. So locks and keys work in much the same way, and so this is where this model sort of came into play. But we understand enough now about entropy and enthalpy in biochemistry to understand that it's a really simplistic and very idealistic way of thinking about things. So if we were to imagine this from an entropic standpoint and we were to think about our enzyme active site, if we were to pre-organize our enzyme active site to have the perfect shape for our substrate key, that would be very unfavorable from an entropy standpoint. Now, when our substrate docks into our enzyme, we can form those intermolecular forces so we can have that enthalpic compensation that will pay that cost. But until this binding event occurs, we don't have that compensation. That compensation happens only after binding. So what we're going to see as we move on to the next slide here is that we sort of have a lock that matches a key, but it's a little bit mushy. We really don't want to pre-organize that shape because that's going to be an entropic penalty that we really can't compensate for. But before we move on to the next slide, let's actually just take a second and talk about the kinds of intermolecular forces that we will see when substrates interact with enzyme. Right? We can have a shape complementarity, right? That has to do with the London dispersion forces. So those intermolecular forces that are not specific, like electrostatic or hydrogen bonding, just sort of the shape and the interaction of these surfaces are going to contribute a lot to the specificity that we're going to see. And so we can sort of see that matched here. And this is a lot of where this lock and key sort of model came from. But once we have substrate dock, we can also have potentially electrostatic interactions. So electrostatic interactions are the interactions of positively and negatively charged species. We also can have hydrogen bonding, right? So a lot of these are going to contribute a little bit more to specificity, so kind of locking in certain residues being close to one another or certain parts of the substrate being close to certain parts of the enzyme active site. But as we've learned before, they really don't contribute a ton in terms of stability. They really have a lot to do with specificity. But again, lots of the same intermolecular forces that we've talked about before come into play when we think about substrate binding to enzyme. So as I kind of highlighted, the reality really is more of an induced fit model to think about substrate binding to enzyme. Substrates that tend to be smaller in size might be a little bit pre-organized, might tend to have a more specific shape, but it's going to be entropically unfavorable to have a very well-defined enzyme active site. So we're going to call this a little bit mushy for lack of a more scientific term. It's not well ordered. However, as the substrate begins to dock into the enzyme active site, then we can start forming that more ordered enzyme active site because now we have that substrate available to provide that enthalpic compensation for it. Just like we saw in our previous slide, we're going to have enzyme then do chemistry and convert that substrate to product, and then we're going to have the release of those products, which actually is an entropically favorable process for the enzyme because it can return to having some entropic freedom and disorganization in the enzyme active site, which is thermodynamically favorable. So some more nuts and bolts before we're ready to get into kind of studying the details of those four enzymes. When we talk about the different modes of catalysis, we're just kind of saying what are the different roles that amino acids can serve. Well, amino acids could serve as general acid catalysts, general base catalysts. So general acid catalysts donate protons, general base catalysts remove protons. Something serving an electrostatic catalysis role is basically helping to stabilize by providing the opposite charge. And then something that's acting in a covalent catalysis role is an amino acid residue that's actually forming a covalent bond. So these are the four main modes of catalysis that we're going to see in the enzyme active sites that we'll study. One of the big skills for this chapter is learning how to detail an enzyme active site. Also might say we're going to diagram an enzyme active site. Now that doesn't mean that you have to draw what you see here, but given a picture of what you see here, I want to describe the flow of electrons and the change of substrate to product that we see here. So anywhere that chemistry is going to happen in our uncatalyzed reaction, we're going to see either amino acids or coenzymes, those vitamins or metals that are going to help perform the functions. So what we want to be able to do is map active site residues onto both the uncatalyzed reaction, so again, those are your reaction mechanisms, identify where they do chemistry, and then kind of map that onto what we see in the enzyme active site. So this is what we see for RNase A, and we'll again explain this in a little bit. But detailing an enzyme active site is really a narrative description of what's going on here. So we're going to treat detailing an enzyme active site kind of like a Mad Libs. So again, the Mad Libs works like this. You're going to say the residue name, acting as A, tell me the mode of catalysis, serves to, tell me the chemistry that it performs. So here's kind of some important details that'll help put that Mad Libs together. Look in the enzyme active site and find the residues that are important for chemistry. They're usually going to be amino acids that have a number next to them. That number just tells you where they are in primary sequence space. So it's really interesting. These two histidines that we see here are very distant from one another in primary sequence space. But when the enzyme folds into its three-dimensional structure, its tertiary structure, these amino acid residues are put very close to one another so that they can serve their specific roles. We're going to want to be looking for nucleophiles and electrophiles, and the arrows that we see in our enzyme active sites are going to help with that. Remember that arrows always start at nucleophiles, so they start at lone pairs or at bonds, and they go to electrophiles, which are going to be atoms. Again, the mode of catalysis is what we saw on this slide. These are the 4 kinds of catalysis we're going to see. And again, identifying the major mechanistic outcome, just tell me what the product is. What happens? Did you make, in this case, a 2', 3' cyclic intermediate? Tell me what's happening in the enzyme active site. So we've already sort of mentioned this when we went through sort of our uncatalyzed reactions, but to just highlight, we tend to see our chemistry happen in the enzymes we're going to study with 2 acts to each play. In the first act, we're going to see chemistry being performed on the substrate. And again, we're going to see a non-water containing hydroxyl group. So again, in the RNase A, we're going to see the 2' hydroxyl of the substrate RNA acting as a hydroxyl. For lysozyme, we're actually going to see an internal nucleophile. I shouldn't say. For lysozyme, we're actually not really going to see a hydroxyl surrogate, but where we had seen water sort of come in and interact with an electrophilic intermediate, we're really going to still see that electrophilic intermediate without water coming in till the second act. And then in serine proteases, what we're going to see is a catalytically reactive serine acting as a nucleophile. So in all of these, we're going to see chemistry on the substrate happen early on. We're going to see cleavage of our linkage, either our phosphodiester, our glycosidic, or a peptide linkage. The second act really is where water is going to come in, and that water is often going to be used to regenerate the enzyme active site. So the water really isn't even being used to do the chemistry that we think it's performing. It's really being used to regenerate the enzyme active site. So like we highlighted in our earlier slides there, the 3 main steps that we're going to see in all enzyme active sites is we're going to perform chemistry on the substrate, we're going to use all of the reagents that we saw in our uncatalyzed reaction, and in this situation it's generally water that we need to make sure that we see, and we need to make sure to return the enzyme to its active state so that it can repeat its reactions. So again, I encourage you, practice speaking this story language with the enzymes that we have to be able to describe an enzyme active site. There's a good chance on your exam that you'll see detailing the enzyme active site for one thing that you've seen, and then having to detail an enzyme active site that you haven't yet seen to show that you can apply these principles to a new situation. Okay. The theme that we're going to do to tackle all 4 of these reactions is we're going to talk a little bit in the beginning about what this enzyme looks like, where we find it, what it does, why we have it, a little bit about some structural details. I'll let you know if there's numbers that are important for you to know. But RNases are enzymes that, like their name implies, degrades RNA. They hydrolyze that phosphodiester bond. So why do we have them? Big picture concept is that RNAs are not supposed to be long-term passengers in our body, right? Messenger RNAs, when we're doing transcription of genes to translate into proteins, messenger RNAs usually are fairly short-lived. Viruses are foreign, so your body is well equipped to recognize RNA as a transient species in your body and has lots of mechanisms to get rid of it. So RNase A is produced by your pancreas, again secreted in the gastrointestinal tract. That kind of helps with maybe viruses that are in your GI tract. But we also talked about the fact that you've got lots of RNases on your hands. And again, your hands always being in touch with the external environment, trying to make sure that we protect ourselves from foreign things like viruses. RNase A is a pretty small enzyme, only 124 amino acids. Some things that are important to know is it's got 2 catalytically essential histidine residues. So again, we've got one histidine that has a pKa that's shifted greater than 6 so that it maintains its protonation state, and it's going to be protonated for catalysis, and then histidine that has a pKa that's shifted a little bit less than 6, so it is going to be then deprotonated to facilitate its role in catalysis. So another important skill is to be able to think about how would a certain environment shift a pKa. Okay, so we have some specific examples here with histidines and RNase A. We're going to see a glutamic acid residue with lysozyme, but just be able to think about this generally. How would this type of an environment shift a pKa value, up or down? All right, so we're going to detail the enzyme active site. This is a busy slide here, but I kind of highlighted in different colors here where we see the chemistry happening. Okay, so we're going to start over here. Always look where arrows start. That's going to help you detail the enzyme active site. So here we have histidine 12 acting as a general base catalyst because it's serving to deprotonate the 2' hydroxyl of the substrate RNA. That's important because it increases its nucleophilicity towards attack on the electrophilic phosphorus of the phosphodiester bond. So that's what happens in this first 2 set of arrows. We've got other chemistry happening here, so we changed colors here to indicate what's happening now. We're going to be cleaving the phosphodiester bond. Cleavage of that phosphodiester bond is facilitated by histidine 119, acting as a general acid catalyst. So it's protonating this leaving group 5' hydroxyl of this downstream RNA. And now we're going to have departure of this downstream RNA, which allows for the diffusion of water into the enzyme active site. So I know a lot of you are just going to try to memorize this language. And again, that's not a bad thing to kind of memorize the Mad Libs portion of it. But try not to just get into the habit of memorizing this. Try to understand what's going on so that you could do this then for an enzyme active site that you haven't seen before. All right, let's move on to the next step. So again, in this next step, we're going to start here. Our arrows sort of start down here with this histidine. So histidine 119, now acting as a general base, serves to deprotonate water, increasing its nucleophilicity towards attack on the electrophilic phosphorus of the 2' 3' cyclic intermediate. Opening of the 2' 3' cyclic intermediate is facilitated by histidine 12, now acting as a general acid, serving to protonate this 2' hydroxyl leaving group. If we think about what's happened now, again, this is sort of a standard ending that you might have. We've got departure of the remaining nucleotide fragment. So reaction on the substrate is now complete. All reactants have been utilized. That is, we've seen water be used, and the enzyme active site has been regenerated. So not necessarily something here that has to be said, but this reminds us of the 3 steps that we have to have happen. Reaction on the substrate. Have we used all reactants? And has the enzyme active site been regenerated? So that's RNase A. So again, a blank one was put up on the board so that we could kind of run through this, have you guys practice. Again, we actually acted this out, right, with our role playing of the enzyme active site. So hopefully that was helpful for you to kind of see the different residues, where they positioned in terms of the substrate, who was donating a proton, who was abstracting a proton, and so forth. RNase A is a pretty straightforward, kind of good one to start with because not a lot of stuff is going on. We moved on then to carbonic anhydrase. So So we first needed to look at sort of the uncatalyzed reaction here. This was actually a reactant mechanism, right, that you had in one of your exams, one that you hadn't seen. But again, water is going to nucleophilically attack the carbon of what equates to a carbonyl here in carbon dioxide, generating an intermediate that we need to do a little bit of proton shuffling around to get to bicarbonate. Right? So this is the chemistry that happens with carbonic anhydrase. A reminder that right now we're looking at it in this direction, which is the direction it would happen in the tissues. It actually would happen in the reverse direction in the lungs, where we'd need to take that solubilized bicarbonate, put it back into CO2 so we can exhale it as the gaseous waste product that it is. So just looking at the uncatalyzed reaction here, we can see that if we had a base, it could help deprotonate water, making it a better nucleophile. And if we had an acid here, we could actually help to protonate this, this intermediate here. So the important part is to think about a way to make water more nucleophilic. So how this chemistry happens in this enzyme active site is we have a metal, a critical zinc residue that's going to bind water in the enzyme active site. So to understand a little bit about how enzymes use water-bound metals, when we bind water to a metal, we're going to have inductive electron withdrawal by the positively charged metal here. So again, this represents coordination of a water to our metal. Again, anytime you want to think about something having a decreased pKa, meaning it's more acidic, deprotonate it and ask, is what's left over stabilized by the environment that I'm in? So again, if we deprotonate water and generate hydroxide, that's actually going to be stabilized by the positive charge in the metal that we have here. So again, the extra electron density that we have when we deprotonate water can be stabilized by the metal. So we effectively take a pKa of 14 that we had for water and decrease it such that we make water more acidic, so more of it can live in the hydroxide form at a neutral pH. So that's a really important way that metals can help, is they can increase the effective concentration of hydroxide without changing the pH. All right, so let's look at this enzyme active site. Right, thinking about the function of carbonic anhydrase, right? It's a reversible reaction that interconverts gaseous carbon dioxide with soluble bicarbonate. We've got this catalytically essential zinc residue that coordinates to 3 histidines. So again, here's the entirety of the enzyme. Here we can see a blow up of the enzyme active site. Zinc is what we refer to as tetracoordinate, so it likes to bind 4 things. 3 of those 4 things are catalytically essential histidine residues. The 4th thing is again a water, and by binding to that metal, it really is effectively a hydroxide. Just a little bit more detail about other ways that metals can be used in enzymes. So any enzyme that requires a metal, we call it a metalloenzyme. Maybe we need some chemistry to happen on a redox front. So maybe we've got iron that can toggle back and forth between iron 2+ and iron 3+. We'll see this a lot when we look at enzymes in the electron transport chain. Those enzymes do a lot of redox chemistry with metals that are going to be able to shuttle and ferry those electrons back and forth in enzymes of the electron transport chain. Sometimes you just have metals that are playing structural roles. It may be divalent metals or maybe just monovalent metals, but sometimes you need them to help maintain structure. Maybe you have a metal that can help bind and orient an anionic substrate. So amino acid residues can help with this, but again, metals are well known to do this. Maybe you also can stabilize or shield negative charge buildup in an enzyme active site. So again, what we're going to see here is metals are really only able to help in stabilizing anions or negative charges, and that's obviously because metals are positively charged. So not a lot to really detail with the enzyme active site. So this isn't going to be one where you're going to detail the enzyme active site particularly. But what we see here is we've got this water-bound, zinc-bound water. So we've got, again, we're just kind of highlighting this. I like this picture better here because it actually shows the different steps. This is the enzyme active site. These imidazoles here represent this imidazole ring on histidine. So again, zinc binding to 3 imidazoles, water effectively in its decreased pKa, so hydroxyl form. So nucleophilic attack on carbon dioxide generates bicarbonate. We're going to see water that comes in here and essentially displaces the bicarbonate binds to the zinc and regenerates that enzyme active site. So again, another situation here where it's sort of a two-act play. We don't see water coming into the second act really serving to regenerate the enzyme active site. So not really one that you don't worry about this one in terms of detailing the enzyme active site. There's not really anything to detail in particular, but I do want to highlight that we do see sort of the same second act appearance of water to regenerate the enzyme active site. All right, 2 more enzymes to get through here. If you want to take a break, this might be a good time to sort of take a break. Return when we're going to talk about lysozyme here. So again, lysozyme breaks glycosidic bonds. It actually interacts with glycosidic bonds that are in bacterial cell walls. So really interesting kind of aspect of bacterial cell walls is they tend to sort of be this 3-ply component of having glycosidic linkages with carbohydrates, peptide linkages, and then disulfide bonds within those peptides. And so we kind of have this network that needs to be broken. And so lysozyme helps to do that. That weakening of the bacterial cell wall actually inhibits the bacteria from growing. And so lysozyme is actually a natural antimicrobial agent that's part of what we call our innate immune system. So kind of this generalized warfare agent. It's not something that acts specifically. It doesn't have a recognition of any specific sort of microbe or anything. It just sort of specifically works to break those glycosidic bonds. So lots of lysozyme that's produced in your tears, saliva, mucus. There's tons of it in egg white. So again, this is sort of an interesting thing. A lot of these proteins that are well studied came from sources that were very easily isolatable. So obviously egg white is very easy to kind of obtain, and a lot of the protein that's in in egg white is actually lysozyme, so very rich in lysozyme. Another small protein, relatively speaking, 129 amino acids, disulfide links, but this is again the critical piece that I want you to know. It's got a pKa that's shifted towards neutrality for glutamic acid. Normally glutamic acid is going to have a pKa of between 3 and 5. So in this case, we're going to have a pKa that's shifted higher, that allows glutamic acid to maintain its proton, so maintain that neutral state. And we talked about this in class. That's because it kind of lives in this nonpolar microenvironment that is better suited to having something that has no charge than negative charge. So another kind of important piece to think about here is that when we have our substrate that binds, so if you think about the substrate here, it's basically a 6 residue section of a carbohydrate. So there's 6 residues that are here. When the 4th residue kind of sits in this enzyme active site, and they label these with letters rather than numbers, so D is the 4th letter, so that's why this is the 4th residue here, it actually becomes a bit sterically hindered. And we're going to talk about that. That's going to be entropically unfavorable, but we're going to see that there's an enthalpic compensation for it. So again, blowing up, I'm just going to kind of highlight here. Here is this residue. So here's this residue that's in this weird half-chair conformation. As we're going to talk about in a minute, the purpose of that half-chair is it allows these 2 atoms to be coplanar, and that's important because if we're going to form a pi bond between these to resonance stabilize an intermediate that we have, or I should say a transition state that we're going to have, we need to have these orbitals be able to overlap. So that's why we have that. So this is entropically unfavorable, so we must have some enthalpic compensation. That enthalpic compensation comes through a few residues, this valine in particular, that helps to sort of hydrogen bond and stabilize that sterically strained half-chair conformation. So again, we have this strained half-chair conformation because we need to have orbital overlap between these 2 atoms in order to stabilize the transition state that we're going to have. So we're going to see that on the next slide. But we're compensated because we've got several favorable side chain interactions here that help to stabilize that. Highlighting some important residues that we see here. Again, that D residue being in that half chair allows those atoms to be coplanar. We're going to have a critically important glutamic acid residue that has a unique pKa shifted towards neutrality, so it's maintained in its protonated state. So we've got that guy there. We've got an aspartic acid residue, and that important aspartic acid residue here, shown right here, is going to allow us to do covalent catalysis. So So this aspartic acid residue has its regular pKa. It's going to be negatively charged, makes it actually a better nucleophile. We're going to see that it's actually going to form a covalent bond with this carbon atom here. So we'll see that in a couple of slides here. And then this valine residue that we talked about, that valine residue, this is actually not the side chain of valine, but it's going to be the backbone atoms of valine that are going to facilitate in having this enthalpic compensation to stabilize this half chair. All right, again, the purpose of this half chair. If we look at this uncatalyzed reaction here, again, we've got this acetal or ketal here. We protonate this guy to make him a better leaving group. Internal nucleophile is going to push out. So we're going to have this lone pair of electrons form a pi bond here to allow this to depart. So we've just kind of drawn this before and not really thought about the fact that, well, hey, if we're going to form a pi bond, these two atoms are sp2 hybridized, which means they need to live in the same plane in order to have their p orbitals overlap. So the geometry in a chair conformer would not allow for that. So we have to adopt this half chair conformer to allow these two atoms to have their orbitals overlap to form this pi bond. So again, we don't see carbocations with incomplete octets, so we don't just have this leave. We use this adjacent lone pair of electrons to help push that out, and in doing so, we also avoid a carbocation intermediate, and we have an intermediate that is stabilized by these adjacent lone pair of electrons forming that pi bond. All right, so we're going to go through the details of this enzyme active site here. We actually have it on two separate slides here because it's too much detail to kind of talk about here. So I'm not going to take the time in this video to go through the details of it, but just to kind of highlight, after our substrate polysaccharide binds, again, we're going to have I don't like that they don't show this here, but this is our half chair conformer. When you show Hayworths and not chairs, you really don't see that, but this is our half chair conformer. And again, we're going to have glutamic acid acting as a general acid to protonate this oxygen of the glycosidic bond. Okay, that is going to allow us to facilitate the cleavage of that glycosidic bond and then the formation of this oxonium ion transition state. Again, I don't like that they show this as a carbocation because it's very unstable, so we are likely to exist in this resonance form where we have orbital overlap between where these lone pair of electrons kind of come down here. So again, in order to have these two form a pi bond, they need to live in the same plane. And so that's why we needed to have this half chair conformer. So again, once we get to this point, we're going to have aspartic acid now acting as a covalent catalyst, interacting with this electrophilic carbon here and forming this aspartic acid substrate covalent intermediate. So again, now we've got an intermediate that's able to be isolated. We have departure and diffusion out of sort of the downstream fragment of our sugar. So now water can diffuse into the enzyme active site. We're going to sort of see a reversal of chemistries here for glutamic acid, now acting as a general base, serving to deprotonate water, increasing its nucleophilicity towards attack on this aspartic acid intermediate here, this covalent intermediate. Nucleophilic attack here is going to allow for displacement of aspartic acid. We've regenerated our enzyme active site. The upstream kind of carbohydrate fragment can diffuse out of the enzyme active site, and we're ready to continue chemistry. So again, one of your homework problems has you thinking about how to detail this. So I'm not going to go through and reread through all of these, but again, you're not going to have to do, the entirety of Lysozyme, for example. You'd have one step that you need to do and kind of comment on. All right, almost to the end here, serine proteases. Okay, so serine proteases, it is a they because it's a class of enzymes that hydrolyze amide bonds in the GI tract, but they have specificities for side chains that flank the bond that's to be cleaved. So again, if this is the bond that's going to be cleaved here, we can see adjacent to it, we've got a specificity pocket. We're going to see more of that on the next slide here. But all of these enzymes, we're going to talk about three of them, three with different substrate specificities. They all have a common catalytic mechanism that involves a reactive serine that's going to act as a surrogate for water. So again, why do we have these different enzymes, right? These are going to be secreted by your pancreas, And they're going to facilitate the digestion of dietary peptides because only those monomeric amino acids can be taken up out of your GI tract and into your bloodstream. So again, all of these enzymes are produced in your pancreas. We'll talk about the importance of producing them in an inactive form. Your pancreas is made out of protein, so you do not want to be making an active protease in your pancreas, otherwise you're going to digest it. A little bit bigger of a protein here, but not that much bigger than we've seen, and still relatively small protein. One of the things to highlight here, a number you don't need to know, but there's very little sequence homology between all of these. So all that's really important is that you have these 3 amino acids kind of in your enzyme active site. We call this the catalytic triad. So we've got a catalytically reactive serine, histidine, and aspartic acid. We'll see those on the next couple of slides here. But again, very different primary structure locations in all of these. You know, we've got 195, 57, and then aspartic acid 102. But the tertiary structure brings them all into the same location, and that allows it to have its function. So even between the 3 different serine proteases that we're going to study and talk about, they all basically put this catalytic triad right in the active site where it's needed. So again, this catalytic triad is a reactive serine residue that's going to act as a water surrogate. It is made more nucleophilic and more basic because a histidine that's adjacent to it is going to deprotonate it. That histidine is actually made more basic by aspartic acid serving to kind of donate electron density. So we sort of have this three-way system here that allows serine to do its important chemistry, and we refer to this as a catalytic triad. So speaking of that substrate specificity, so again, if this is the bond that's to be cleaved right here, there's 3 different classes of side chains that we can see for adjacent residues. We can see bulky aromatic side chains like phenylalanine, tryptophan, or tyrosine. That's going to be a substrate for chymotrypsin because the adjacent substrate binding pocket is a slit-like hydrophobic pocket that aromatic residues can fit in. Trypsin, on the other hand, has a substrate binding pocket that has an aspartic acid that's buried in the bottom that'll form a very favorable electrostatic interaction with cationic side chains like lysine and arginine. Elastase, on the other hand, has a very crowded binding pocket, so it's going to be a substrate for small neutral residues like alanine and glycine. And again, the reason for that is big things aren't going to fit in there. So a question that we spent some time talking about in class, and I'll revisit just briefly here. So why doesn't something that has an alanine or a glycine be a substrate for trypsin or chymotrypsin, since clearly it can fit into this? So we were reminded that in order for any of these residues to fit into these enzyme active sites, they're probably accessible to water, and so there's probably water that's surrounding here. So we have to kind of remove that water, and that's going to be an entropic penalty or an enthalpic penalty, and so now we need to compensate for it in some way. And so we compensate for it in this very deep binding pocket that has something negative in the bottom by having lysine or arginine. And again, for chymotrypsin, we've got this slit-like pocket that's going to form intermolecular forces with these aromatic residues. We're not going to be able to do that. We're too small to make these interactions or to be able to fit in that slit-like pocket. So we can't have that enthalpic compensation with these small residues. So just like our basketball and baseball analogy with our potassium channel, we see the same sort of thing here. All right, so serine proteases are another one that if we look at the details here, it's a fairly lengthy script that we have here. So I'm not going to go through the details of the script here. I'm going to kind of highlight some important points here. I will sort of go through what we did sort of in class just so we can see all of these, but I'm not going to follow along with the script per se. So again, we've got our substrate that we're going to have bound here. And again, our substrate looks very small, but remember, all we're showing is the important catalytic atoms here. We're not really showing any of the side chains or anything here. And then again, we've got our catalytic triad here. So again, what we're going to have to start here is our serine is going to nucleophilically attack our substrate polypeptide, forming a tetrahedral intermediate. The nucleophilicity of serine is enhanced by histidine 57 acting as a general base, and his basicity is enhanced by aspartic acid helping via electrostatic catalysis. So at the end, we form a tetrahedral intermediate, and what's not mapped on here but shown in blue here is, remember, we've got this oxyanion hole. So it's a positively charged pocket that can stabilize this tetrahedral intermediate that has a negative charge on that oxygen atom. So anytime we form a tetrahedral intermediate, we tend to see decomposition of the tetrahedral intermediate. Now if we break this bond right here, that just gets us right back to product. In this case, we're going to be breaking this bond here. If we want to have something be a good leaving group, the best thing to do is to protonate it. So now we're going to have histidine 57 acting as a general acid to protonate the nitrogen of the N-terminal peptide so that decomposition of the tetrahedral intermediate happens easily to break the peptide bond. So in this case, we now generate an N-terminal fragment that can diffuse out of the enzyme active site, allowing water to diffuse in. But we got a problem here because serine is still attached to this other portion of our peptide. And even though the rest of our catalytic triad is kind of back in its active state, the important serine is not. So water diffusing into the enzyme active site, we're going to repeat this process again. So even though these two guys have been returned to their active states, serine has not. And remember, the purpose of water in almost all of these enzymes is to regenerate the enzyme active site. It actually doesn't do the initial chemistry. So we're going to have nucleophilic attack by water on the carbonyl carbon of this serine peptide intermediate here. The nucleophilicity of water is enhanced by histidine 57 acting as a general base. And again, its basicity is facilitated by aspartic acid acting as an electrostatic catalyst. So once again, we generate a tetrahedral intermediate that's stabilized by our oxyanion hole. And then we're going to decompose this tetrahedral intermediate. This time we're going to break the bond to serine, and departure of serine is facilitated now by histidine 57 acting as a general acid to protonate the serine's oxygen to generate a better leaving group. At this point now, we now have our new peptide here, our C-terminal portion of our peptide that can now exit. We've regenerated our serine. Our serine is now free. Our histidine is back in its deprotonated state, which is its native state, and aspartic acid, again being solvent inaccessible, was just kind of hanging out there doing its thing. So fairly detailed one there. That one took a little bit longer to act out. We needed more players because we needed people to be the enzyme active site, so we needed an oxyanion hole. We had 3 residues here participating in the enzyme active site, so lots going on with serine proteases. The last thing to sort of talk about here, so again, here was the oxyanion hole, just kind of showing the structure. We've got these backbone atoms that can help stabilize something that's negatively charged, like this tetrahedral intermediate. The last thing that we just sort of talked about here was sort of this dilemma that we have when we in our body are making proteases. Our body is made of protein, so it's really important that these don't become active where they're not supposed to be active. And so 2 situations sort of help here. We certainly don't want these to be active in our pancreas where they're being made. And so we make them in an inactive form where we've got an N-terminal domain, just a little 14 residue N-terminal portion here, that if this N-terminal portion is here, the whole thing sort of misfolds so that we don't put that catalytic triad together, and this isn't active. But what's really interesting is that we've got a lysine here at position 15. So if this protein is made and then misfolded in the pancreas, as it makes its way into the GI tract, it encounters either active trypsin or other peptidases that live in your GI tract, and that basically is going to cleave off this 15 residues here. So it's going to cleave this bond right here. It's going to cleave off this N-terminal domain. The rest of the protein can sort of refold. We can slide that catalytic triad together, and now we've got active trypsin. So this is again what happens in your GI tract to make sure that this protease is not active until it gets into the protective environment of your GI tract. Remember, even though your GI tract really is made of protein, it's got a very thick mucus layer that helps to protect it. So some terminology just to kind of highlight here. is anytime we produce something in an inactive form, we call that a proenzyme. So when we talked about insulin, we talked about making proinsulin. That was a larger version of the enzyme that was in inactive form until we got rid of that C peptide. So specifically for these proteolytic enzymes, we call the proenzymes zymogens. So a term I want you to know, zymogens are the proenzyme or larger inactive forms of our proteolytic enzymes. They actually do have some specific names as well. And again, I do want you to know trypsin, chymotrypsin, and elastase, and I want you to know their substrates that they're specific for. But the zymogens are called trypsinogen. So trypsin, we're going to add the ogen to the end. So trypsinogen and then chymotrypsinogen. So just kind of remember that we add the ogen at the end to make the zymogen form. The last thing just to kind of highlight here is, well, not only do you not want these proteases, becoming active in your pancreas where they're made, we also don't want them to be active if for some reason they happen to get out into our bloodstream. So within your bloodstream, you actually have a trypsin inhibitor protein. This is actually called BPTI because it's bovine pancreatic trypsin inhibitor. So you have a human analog of this, but this is a protein that binds so tightly to the trypsin active site that if you happen to have had trypsin get out into your bloodstream, it would find some trypsin inhibitor and bind to it and then basically have it be out of commission. So even though this is technically an inhibitor, it binds so tightly and so irreversibly that we call it an inactivator. So those are terms that we'll actually expand on more in our enzyme kinetics chapter. But this represents the end of our enzyme catalysis chapter. Wow, guys, sorry, long video there, probably going to be something you're going to want to watch in a couple of parts.